So hello, thank you for the opportunity to talk today. I'm Parmveer Delay, an assistant professor in gastroenterology at the University of California, San Diego. So I'll be talking about the US IBD Health Outcomes Consortium and some real-world approaches to studying biomarkers for inflammatory bowel disease. As a brief background on health, about 4 to 5 years ago, a group of us came together with the goal of trying to generate real-world evidence. This is a multi-center, multinational consortium of over 20 sites spanning North America. And the goal was to generate real-world evidence for emerging therapies and strategies for treating inflammatory bowel disease. The ultimate goal was to be able to help optimize the integration and validation of effectiveness, safety, and use of these therapies, but also strategies as they relate to the use of biomarkers in clinical practice. What we wanted to set out specific Research Alliance was to get a good mix of community and academic centers. The biggest limitations with research and clinical practice is that the tertiary referral bias ends up creating differences in patient populations. So we wanted to make sure we had academic centers who saw refractory complex patients with multiple prior therapies, but also community centers who are seeing more often treatment-naive patients early in the disease course prior to the development of complications. This would allow us to help select the appropriate patient populations, and this was really most pertinent as we began to think about how we would use biomarkers and study biomarkers in the future. The platform and design of the consortium is complex in its background and how we put it together. First was the data part that we needed to bring together. So we had to set up data sharing agreements between all of the centers, which allowed for data capture and data transfer in a seamless process. This was probably one of the biggest background hurdles to make sure that every institution's security protocols were followed. Each institution was then responsible for their own data collection. This helped maintain local site security for data collection and helped make it a little bit easier for transfer and integration once everything was completed. The data was then de-identified by each site and sent to UCSD, which was a centralized data repository. This then formed the consortium of data that we worked from over the past few years. The data was recoded in-house, QC checks were done, and it was compiled and analyzed with multiple different specific outcomes that we were focusing on. We began the consortium with a big focus on effectiveness and safety for biologics and small molecules that were emerging in clinical practice. Over time, we began to evolve towards looking at comparative effectiveness, particularly when we consider biomarkers and how they can be used for comparative effectiveness. And then we eventually transformed into also wanting to study predictors and personalization of therapy. As it relates to biomarkers, you'll see a few examples of how we've been able to do that throughout this talk. These are just a few examples we had. Our very first initiative was the Victory Consortium, which studying the effectiveness and safety of vedolizumab for ulcerative colitis and Crohn's disease. So over the past 4 years or so, we've had multiple publications documenting the effectiveness and safety of this drug in clinical practice. And this was our first sort of validation of the consortium as a whole, to be able to bring data together from these centers, to be able to study this in a coherent and consistent manner, and to be able to provide evidence that was consistent with what was being achieved in regulatory-based clinical trials. What we wanted to do from there, though, is we really wanted to expand on this and begin to think about how we could integrate both clinical and biomarker-based measurements of disease activity to guide treatment decisions for these biologics as they came to practice. So our first attempt at this was building a decision support tool for vedolizumab use in Crohn's disease. What we did was something rather unique, which had not been done before in the field of IBD. We actually linked together the regulatory clinical trial data sets from the phase 3 clinical trial program with the routine practice data sets that we had collected across the Victory Consortium. So what we took was the phase 3 Gemini 2 randomized controlled trial data sets. We looked at outcomes of clinical remission, steroid-free remission, and durable remission, which were primary and secondary outcomes in the trial that led to approval of the drug. We did different modeling approaches, but we ended up coming up with a set of common predictors. Several of these were just basic clinical variables that we know to be important, but 2 of these could be considered lab-based biomarkers that we were applying in a unique perspective, which were baseline albumin and baseline C-reactive protein. So what we did then is we needed to harmonize the Victory Consortium data to be able to not only integrate the clinical variables, but also the biomarkers that were being collected across the institution. This was our next big hurdle in terms of data harmonization. But what we benefited from was the fact that CRP and albumin were common biomarkers that were available broadly across all the institutions and were being used in a very consistent manner across each lab for disease activity assessment and disease monitoring. And so we were able to rather easily and seamlessly harmonize the data capture for these biomarkers in routine practice to be able to use the consortium data set for validation of this combined clinical biomarker-based decision support tool for prognostication. And here are the results from that validation study, which was published a couple of years ago. And what you can see here at the top are the 5 variables that we included, the last 2 being lab-based biomarkers of prediction of response to vedolizumab in Crohn's. And what you can see here in the bar plot is when we stratified patients in this routine practice consortium with the combination of these clinical and biomarker tests, we could get a very clear separation in identifying patients who would or would not respond to therapy. The orange bars were patients who were predicted to have a low probability of response for the 5 outcomes of clinical remission, which is CREM, corticosteroid-free remission, which is CSF-REM, mucosal healing or healing of the intestinal lining, MH, and then deep remission and corticosteroid-free or deep remission, which is a combination of clinical and endoscopic outcomes. And with this combined clinical biomarker diagnostic tool, we were able to identify with 100% sensitivity patients who would not respond to this drug and therefore would likely be most appropriate for alternative therapies, and we're able to prognosticate differential rates of response in these patients. And this was our first proof of concept and proof of ability to actually use biomarkers and validate them in clinical practice for use in patient care. We then went on to actually validate this internationally. There's several other groups across the world who do consortium-based research in IBD. One of them is Jetaid in France. So collaborating with these investigators, we were actually able to take this clinical biomarker-based decision support tool and show that we could predict outcomes for vedolizumab in Crohn's disease in another data set that's located outside of the US. And again, we benefited from the fact that labs have consistency in the use and testing and standardization of these biomarkers, which are widely available across institutions, which allowed it to make it easier for us to actually study and validate this biomarker-based decision support tool in practice. And this was a rather simple effort, but it was an important effort for us to be able to understand how we could begin to get into studying biomarkers and identifying biomarkers that prognosticate disease outcomes and treatment outcomes in routine practice. So that led to the second part of our consortium efforts, which were to begin to understand how we would create common central IRBs and resource support. We wanted to begin to think about how we could create a common lab for blood and stool assays and testing, along with standardized shipping protocols that ensured integrity of these tests regardless of where they came from across the US. And we've now begun to think about how we can really centralize and standardize the gold measures of disease activity upon which these biomarkers might be used to study treatment response or prognostication. This has benefited from partnerships with both industry sponsors like Takeda, who funded the Victory Consortium, and several other industry sponsors like clinical research organizations that help with endpoint development and centralized reading. And finally, the core of this is benefiting from master protocols that we've been building, which allow for seamless integration or data stacking across different platforms. And what you can see here on the far right of this under the master protocol is if you can create these umbrella protocols for Crohn's disease and ulcerative colitis, you can actually stack different levels of data to understand specific therapies, care pathways, and begin to get into the potential for studying biomarkers within patient-level randomized trials or larger cluster randomized trials without large interruptions that can benefit from routine care and standard of care follow-up processes. So our first example of this, and our first successful attempt at doing this, was actually studying hyperbaric oxygen therapy for ulcerative colitis. So hyperbaric oxygen therapy is the delivery of high amounts of oxygen into the body to relieve hypoxia, inflammation, and and ischemia, which are chronic hallmarks of inflammatory bowel disease. So we embarked on 2 separate phase 2 clinical trials to study the efficacy of this therapy in hospitalized acute severe ulcerative colitis patients. Although our primary aim was to study efficacy, we did want to embark on studying biomarkers of mechanism and biomarkers of treatment response. And because we had built this infrastructure within the consortium for data integration and created now a standardized protocol for sample collection across the sites, we were able to actually get into single-cell sequencing and multiomic microbial sequencing using a standardized centralized lab at UCSD, which followed a very clear protocol and allowed for harmonization of these biomarker assays and assessments across centers. The trial itself was successful. This was published in 2018, and I won't go into this too much, but it's mostly just to show that the efficacy was there, and therefore there was some value in beginning to think about how we could study biomarkers of treatment response or differential response in this patient population. So the blue bar is the reduction in activity or disease activity with hyperbaric oxygen therapy versus the change seen with the sham arm in this double-blind randomized trial. When we began to look into biomarkers of treatment response, one of the things that we piloted here and were successfully able to do was a standardized tissue collection process across all the centers so that we could actually do single-cell sequencing. And so single-cell sequencing is being able to identify a specific cell subtype, sequencing that cell subtype specifically to understand how the immune signature changes with specific therapies or interventions. This was one of the first efforts to be able to do this within a randomized clinical trial setting. But because we had set up these master protocols and standardized protocols for collection, shipping that maintained integrity of samples, we were able to create uniformity across the sites in this, investigator-initiated study. And what we specifically benefited from was the ability to lump this or add this onto standard of care protocols, which really helped optimize feasibility. This is a visual image of our ability to study neutrophils specifically versus other CD45 positive immune cells, and to be able to differentially select out some of these immune cells to look for immune cell-based signatures of treatment response. A secondary outcome that we also wanted to look at was actually the microbiome. And so because UCSD has a robust microbiome core facility, and we had standardized sample collection down to the type of container that was being used, the type of collection process, storage temperatures at local centers, we were able to do multiomic sequencing across all these patients. So we were able to do not only serum and fecal proteomics, but also metagenomics and 16S, as well as metabolomics and peptidomics, to allow for a multi-omic understanding of changes in the microbiome and its metabolism that occur with hyperbaric oxygen therapy. One of the biggest things with the biomarker research is that, what we wanted to do was really just store everything and then look to come to it afterwards all at once, which allowed for reductions in any variability in freeze thaw or complications that might arise or data variability that might arise from multiple runs. And we did that, and it's been sort of successful so far. And we've seen some results that have been validated in other studies and some follow-up experiments. And so when we think about keys to success for studying biomarkers in IBD in routine practice, the 3 things that I would say that helped to really define the success for the consortium as a whole are the first being that we had really strong core data sharing agreements. That was one of the biggest lifts that occurred early on, was to be able to get data security, data integrity, and sharing agreements in place across the centers, which became the framework upon which we could build common protocols that were approved locally by each IRB, thereby avoiding concerns about people outside of the institution accessing data and then having unified data collection approaches. When we began to look at biomarkers, we were able to leverage common lab biomarkers because they're standardized across institutions. CRP, fecal calprotectin, albumin, other biomarkers that are common lab-based tests are actually readily easily addressed or studied because we know that each lab follows standardized protocols for these biomarkers and assays that have been validated and studied for multiple other indications. And as we began to think about how we wanted to look into biomarker discovery and begin to think about how we could look to identify new biomarkers of treatment response, prognostication, or disease activity assessments, we really started with building a very strong standardized and centralized sample collection process. which everybody had to adhere to. And then we had a standardized and centralized set of analyses for these biomarker discovery processes. You know, what we were able to do here, that was, I think, probably the reason why we were able to accomplish this with a relatively small budget on an investigator-initiated study, was that we leveraged standard of care practices for collection. We are constantly collecting stool, blood, and mucosal tissue from these patients for routine care. And so to collect an additional sample or to collect a small amount of that would have otherwise been discarded makes it much easier for centers to be able to do this in a real world setting without the need for large infrastructures or large amounts of funding to get this accomplished. And then once it's stored, a lot of biomarker assays are very flexible, and they're really not sensitive to temperature changes and sort of protocols or processes for sample collection. And so you can begin to do a lot of this later on once you have all the samples together to optimize efficiency in some of this work. And so that's just a sort of roadmap of how the consortium has put some of these things together. Some of our early successes in studying CRP as a biomarker and our sort of platform for how we plan to study these biomarkers going forward, particularly within phase 3 or phase 2 and phase 3 clinical trials that occur in the routine practice setting. So thank you for your time today.